Good morning, centers. Good morning. How are you? Itanagar, Shillong, Chuachampur, Senapati, Tamilnad, how are you? All? Good. How was your test? How was the test on Saturday? This is the same question I'm repeatedly asking you on Mondays. Isn't it? So how was it? How was your test? Good, bad? Nothing? No response? Okay. I guess it was difficult. Not good. Why? It was easy paper. Why it was not good? What happened? What were the issues? You don't ask questions from the tests. If you're not able to sort out anything, you're not asking those questions. Yes, everybody. I'm talking to all the centers. Shillong, Itanagar, Tamilnad. Yes. No? Okay, fine. So what were we doing in the last class, Tuesday? We were discussing something. What was it? Yes, Shillong. Both the sections. Tell me. Yes, Senapati, good morning. Yeah, tell me why was test not good? What was the point? What was the part you were missing? You were missing something, or was the test really very difficult? I guess test was easy since now you have to prepare yourself for. Yeah, I guess we did all the lipids. Everything in lipids we have done. We have to start with a new biomolecule today. So we are left with carbohydrates, primary, secondary metabolites, enzymes. Isn't it? That is what we are left with. We start with carbohydrate. But first tell me what was the issue with the test. Why it was not good. Now if you are having revision tests and you are not performing good, you are not satisfied with your performance, we need to discuss it. Isn't it? Don't you feel so? Yeah, everybody. I'm not talking to one center, but I'm talking to each and every center. Senapati, Shillong, Itanagar. I can see 3 centers logged in. Yes. Tell me. What was the issue? This was a revision test and the chapters were very basic. Tell me, children. No problem, no issues? How is it possible? Okay, fine. I guess you don't want to discuss the issues. But yes, I would be talking to you on the WhatsApp group. Let us discuss those things also. Okay, fine. So, shall we start with carbohydrates? Oh, how are you now? Child at Itanagar, how are you child now? How are you now? Get well soon and start preparing. No worries. Just get well soon and okay, fine. Take care of yourself. Get well soon and then you can start the studying part. But make sure you are having those backups. Whatever test you are missing on, you just have those copies from your coordinator so that you can attempt at home at least and can give a result. Because this is what you are coming here for. Okay, fine. Yes, Tamilnad. How are you? Good morning. Okay. So I am going to continue since you don't have any doubts for the test, I guess. The test was doubtless, but still it was not good. I don't know how it could be possible. You don't have doubts. At the same time, The test was not good. Okay. I guess you did not study. Was it? Lack of revision? Okay. So let's now continue with biomolecules. And yes, after today's class, we will complete with biomolecules somewhat, like I'm supposedly going to complete this, and we will have a discussion round about it, about what you have got after this whole discussion, whole chapter, 6 hours of chapter. Okay. So let's proceed with biomolecules. So first and foremost thing, what you know about biomolecules, we have all discussed. We have 2 classes already in the last week about biomolecules, and we have a good background about it. So whatever things you are getting in your system, organic molecules, they are said to be the biomolecules. So we classified it into 2 categories, the biomicromolecules and biomacromolecules. Micro, the smaller ones, and macro, the larger ones, bigger ones. So we decided the larger, smaller thing based on their molecular weight, isn't it? What was the molecular weight for being a biomicromolecule? Yes. What was the molecular weight for biomicromolecule? Yes. Quickly, come on. What was the molecular weight for biomicromolecule? Everybody, Tamilnad, Itanagar, Shillong, Senapati, quickly answer me this question. Everybody. Exactly. Very good. So it was supposedly what answer I got from one of the sections of Shillong is 10,000 Dalton. Okay, so this was basically less than 10,000 Daltons. Okay, so under biomicromolecules, what we studied was, we studied amino acids, we studied fats, or better we would say lipids. We are yet to complete the nucleotide section and the monosaccharide plus the oligosaccharide section. And under macromolecule, what we have is, what are biomicromolecules? Obviously the polymers of the above. What were the polymers? Amino acids, they form a polymer called as proteins. Then lipids, obviously lipids, they exist only as micromolecule, never as macromolecule, so we don't have a macro of it. Then we have nucleotides, they exist as nucleic acids, what we call is DNA. Or RNA. Then the polysaccharide one, which is the polymer of what? Monosaccharides and oligosaccharides, called as polysaccharide. So this was what we were discussing in the last class. We discussed amino acids, we discussed lipids, and we discussed proteins, and the remaining things are yet to be discussed. So we'll start up today with nucleotides. Yes. Can we start? Do we have any doubts on these 3 sections, what we completed in the last class? No doubts? No study, no confusion. Is it? The simple mantra. Okay. Everybody has settled? Okay. So any doubts on these sections? These sections? Do we have any doubts, children? No doubt? No doubt? Sure? So let me ask you some questions. Okay, first question. Okay, you all are in one section today. Okay, why? Are we less people today? Okay, computer is not working. Okay, fine. No issues. The writing is readable? Can you read the writing? Is it very small? Just let me know. Increase the font size or... Okay, answer this question without Googling. Don't Google your answer. Without Googling, answer this question. This is question number 1. Let's see who is going to answer first from the 4 centers we are having. Yes. Second question. Can you read the question, everybody? Yes, children. Quickly answer me both these questions. Very good. Good. Tamil Long, yes. Senapati, Itanagar, yet to answer. These are the questions what I taught you in the last classes. Quickly answer me these. We don't have much time. We have to start the next topic. Yes. What is taking so long? It is so they are so easy questions. These kind of questions you are going to get in your NEET. You won't be getting, they won't be asking you to draw a structure of lipid or draw a structure of hemoglobin. Isn't it? They're not going to ask you very difficult questions, but they're going to ask you very easy questions. See, in the last class what I told you is that quaternary structure of protein is represented by hemoglobin. But hemoglobin and insulin, both of them represent the quaternary structure of protein. So the correct answer for this question would be what? The correct answer for this question would be both A and B. So this is the correct answer for this question. Okay? The lipids found in abundance in the brain, yes, we did this also. It is called as glycolipid. Glycolipid is in abundant and the constituent is sphingolipid. This must be the word I taught you. So sphingolipids, they are basically going to form a structure in association with glucose part or polysaccharide part to form the component of the actual component of your brain, that is called as glycolipids. So sphingolipids and glycolipids, both of them are correct answers. So if you see any of one of them, you have to mark it correct. But yes, obviously, if both options are given, you have to go for sphingolipid. This would be the most appropriate. And sphingolipid what? Sphingomyelin. Done? Clear? Moving ahead with nucleotides. So what do you know about nucleotides? You tell me first what do you know, then I will add it too. Yes, tell me, what are nucleotides? What are nucleotides? Come on. What are they? Are they building blocks of nucleic acids? Yes, they are. What they are is, they are the building blocks of nucleic acids. Obviously. What nucleic acids? They are DNA and they are RNA. So can I call them to be the basic of the genetic material? The components forming a second call. What are they? They are called components forming the genetic material. So whatever genetic material we are having inside our cells, they are basically the monomers of nucleotides. The monomers of nucleotides which are composed into polymers, and they are going to form what? They are going to form the nucleotide part. Sorry, nucleic acid part. So first and foremost thing, what are types of nucleic acid you know? Types of nucleotides you know, sorry. What are types of nucleotides? Obviously, they are ribonucleotides and deoxyribonucleotides. So types of nucleotides are ribonucleotides seen in your RNA, and they are deoxyribonucleotides seen in your DNA. Isn't it? So RNA, DNA, both of them, they have different types of nucleotides. Why? We will see the reason now. So if you see, done with this slide? Sorry, I did not ask you. Done with the slide, children? Yes, this slide. Okay with the slide? Any doubts? Itanagar, Senapati, Tamenlong, Shillong? No doubts? Okay, fine. Copied? No, no copied. Not copied, yet to copy. Please copy fast. Okay, nice. Proceeding ahead. So what are the particular building blocks? Let's bifurcate and see gradually and gradually. So nucleotides equals to nucleosides plus phosphate. Isn't it? Nucleotides is equal to what? Nucleotide is equal to nucleosides plus phosphate. And what is nucleoside equal to? You are going to fill it up. Tell me. Quickly tell me. Quickly. Very good. So it's equals to nitrogenous base plus sugar. Isn't it? When you add nitrogenous base with sugar, I will remove this thing. You get is what? Nucleoside. And what kind of sugar? Is this simple sugar what we eat at home? That you put in tea, in your milk, in different sweets. Yes, Durga Puja is approaching. So lots of things to eat now. Isn't it? Do you celebrate Durga Puja at your place? No? Yes? Okay, fine. Yes? Okay. Nice. So you celebrate Pujo? Good. Okay. Oh, you need holidays. Okay, when you get, let me also know, I also do need holidays to celebrate Durga Puja. Okay, fine. No issues. So Ashtami is on, I guess, Sunday, isn't it? Ashtami is the main puja as far as I know. If you celebrate Durga Puja, and Dashera would be automatically a holiday. So Ashtami falls on Sunday, so automatically you people will be having a holiday about it. And Dashera, I guess, must be a holiday. I don't need to confirm although. But as far as I know, it should be a holiday, Dashera also. I'm not confirm about it. I'm equally naive as you are. So once you get schedule, I will be also getting schedule and I will let you know if it is a holiday or not. Okay, fine. So no problem. You can celebrate Ashtami. It's on Sunday without any problem. Okay. So nitrogenous base plus what sugar you need? What kind of sugar? You need is pentose sugar. Okay, pentose sugar. So which pentose sugar you need? Any idea? So if you're talking about the variations of pentose sugar, I have two pentose sugars. It can be either ribose or it can be deoxyribose. Okay, so you have two of the pentose sugars present in your nucleosides or tides. That is, it can be ribose sugar or it can be deoxyribose sugar. Let us see the difference between both of them. See, if you talk about ribose sugar, this is a ribose sugar. It's a pentose sugar. First of all, pentose means it has 5 carbons in its skeleton. Okay, so this is the basic structure of the ribose sugar. And obviously, if you need to know more about it, this is the first carbon, second carbon, third carbon, fourth carbon, and we have over here is CH2OH, the fifth carbon. So what we do is that we put a prime mark over it. You must have heard 5' to 3', 3' to 5', isn't it? I will tell you why we are putting a prime over it. So if you are having ribose, I would be having at my 2' position OH group. And if you're talking about deoxyribose, what I will do is that I will remove this OH. I will replace in fact the OH with H. So ribose is what? Ribose is basically the sugar where the OH is present on the 2' position. And if you are removing, that is deoxy, D means without, oxy means oxygen, and if you are removing the oxygen, so what you are doing is you are removing oxygen from the 2' position. See, if you are removing the oxygen from the 2' position, what you get is the particular molecule called as, nice color, what you get the particular molecule, typical molecule, it's called as deoxyribose. Clear? So the deoxyribose is a component of DNA and ribose is a component of RNA. So you won't find ribose sugar in DNA and you won't find deoxyribose sugar in RNA strictly. See, that is why we have made here this thing, ribonucleotides and deoxyribonucleotides. The reason behind is what? They have ribose sugar and these have what? They have deoxyribose sugar. So this is going to form the component of DNA and this is what is RNA. Clear? That was a particular thing we are actually demarcating. Done with this? Everybody, Shillong, Tamilong, Senapati, Itanagar. Nice? Clear? Yes, children. Done? Okay, very good. Understood? Clear with the things? Now, let us make the pentose sugar and nitrogenous base attachments. First of all, we'll come to nitrogenous base. So, we all know what a nitrogenous base. Obviously, nitrogenous base are the molecules which are having a basic nature along with nitrogen. That is why we are calling it to be nitrogenous base. So, when you talk about nitrogenous base, what are typical types of nitrogenous bases you have? Again, we will classify into two categories. What are they? It can be either a purine or it can be a pyrimidine. The writing is legible, everybody, all the centers? Tamilong, Shillong? The writing is legible to all? Senapati, Itanagar? Can read it nicely? Or shall I increase my font size? Okay, fine. So, purine and pyrimidine are the two types of nitrogenous bases. Purine has a two-membered ring and pyrimidine has a single-membered ring. So, what we have is, we have purine and pyrimidine bases, two-membered ring and any doubts? No doubts? Everybody? Sorry. Okay. So, two-membered ring and one-membered ring. So, the typical members of purine, yes, who is going to tell me this? First, let me see which center is going to answer. Members of purine. Types of purine you know. Quick. We don't have time. Quickly tell me the types of purine you know. Easy, very easy. No? Nothing? No idea. Types of purine. Yeah. Itanagar, Shillong, Senapati, Tamilong. This is like, if this kind of question comes in your NEET, yes. Huh? Since when? Yes. Other centers? Types of purine. Just take it out, Shillong. I don't agree with your first answer. Just take it out. Recheck. That is right? Yes. Very good, Itanagar. The other type? Yes. Very good. Yes. Now it's correct, Shillong. Very good. Okay. Nice, Itanagar. Tamilong? Types of purine? Okay, which slide you want to see, Tamilong? Sorry, I missed your message. I'm sorry. Which slide? Do you wish to see? First, second? Just see this. This slide? Really sorry, I missed it out. This slide, Tamil long. Okay, just copy please. Okay, in the meantime, rest of the centers. Yes, Senapati. Not answering. Answer quickly. Types of purine. In the meantime, type the types of pyrimidine also. Those who have given the names of purines, they can go ahead with types of pyrimidine too. Yes, types of pyrimidine. Tamilong, just let me know when you're done with this copying. Yes, other centers, quickly answer the types of pyrimidines you know. Nice. Yeah. Done? Yes. And what, Shillong? Very good, Tamilong. This type of purine you are telling me, isn't it? Tamilong? Types of purine you are telling me. Yes. Shillong, you need to add one more base. Pyrimidine, you have one more base. Yes. Okay, very good. All of you, very good. Keep it up. So let's see once purine. They have two types of bases, sorry. Adenine and guanine. Very good. And we have three types of pyrimidine bases. That is, the first happens to be the uracil. Then what we have is thymine and cytosine. Now out of this, what happens is uracil is only present in RNA. It's absent in DNA. And thymine is absent in RNA and only present in DNA. Okay? Clear, children? Huh? Clear? Yes. Why there is only one child always running towards the computer? Why others are not? Huh? Okay. Is he the most active person in the class? Types of cytosine. So if we move ahead, let's see what this is going to proceed with. So once we start with the nitrogenous bases, let's add on and keep on adding. Okay. So let's start up with the nitrogenous bases first. So what we said, nitrogenous base when added with what? When added with pentose sugar, it can be either a deoxyribose or a ribose. What does it give? It gives a nucleoside. And when you add phosphate to the nucleoside, what it gives is, it gives a nucleotide. Isn't it? The basic structural formula. Did everybody? He's energetic? Why and why are you not? Huh? And why? That means, why is he only energetic? Why not you? Okay, fine. He's proactive, I guess. Super active. Okay, nice. Very good. So what we have is, just now if you can see, this is what is nitrogenous base, purine and pyrimidine. So what we started is, we started a particular kind of equation. This slide is clear to one and all? You only answered me these questions, so it must be clear. Okay? So moving ahead. So what you have is nitrogenous base. You're adding pentose sugar. What you get is nucleoside. Okay? And that gives, when you add with phosphate, what you get is the kind of equation we are going to build up with nitrogenous bases. First and foremost, if you take adenine and you add it what you get is adenosine. When you add phosphate to it, what you get is adenylic acid. Next, guanine. Pentose can be anything. It can be ribose or deoxyribose. It is guanosine. And we have is guanilic acid. Third we have is, let's start with the pyrimidine bases. Cytosine, pentose, cytidine, phosphate, cytidylic acid, thymine, pentose, thymidine, phosphate, thymidyllic acid. Uracil, pentose, uridine, add phosphate to it, what we get is uridylic acid. Yes, you have to remember, but once you learn one of the formulae, you're going to learn all of them. So it's not that difficult as seems to be, and all of them are present in your NCERT. It is present in your NCERT. I'm just giving you an equation, just forming an equation out of this. Till now, all the things are from your NCERT, either from your class 12 section or your 11th section. So I'm not teaching you till now anything outside NCERT. This is all sorted out. The NCERT language has been sorted. Yes. Senapati, Shillong, Tamenlong, Itanagar. Any doubts up till now? Any doubts? Let me know, please. If any. No? Okay. Then copy, please. So done with 11th standard, isn't it? This is the last chapter I'm doing with you from 11th standard. I will be starting with human reproduction in the next class. Shall I? Okay. I'll start with human reproduction in the next class. Isn't it? Not left with anything? Read almost everything from this section. Yes. So we'll start with human reproduction in the next class. Copied everybody? I guess we have copied because copied everybody. Senapati, Shillong, Tamenlong, Itanagar, Tamenlong. What is going on at Shillong? What is it? Yes, the girl at third bench. What is going on, child? What are you doing? What is going? Yes, you only. What is going on? The child in green top. What is going on? Let everybody study. Don't talk now. I will be giving you a break after some time. You can have fun at that time and after, obviously, in the lunch period also. Yes. And there is something also going on at last at the back bench. Okay. So we have done copying and let's proceed further. Okay, understood everything? So now let's make what is the particular section, what is the particular structure of this molecule going to be like. So I will be drawing one of them for you. So what happens is, first position. Yes, there was a question, isn't it? I asked you a question, why do we mark 1, 2, 3 prime over here? What's the use of marking prime? The first position has what? The first position has a base. That is a nitrogenous base. Isn't it? This is a particular sugar. Let's mark it to be, since I will be drawing a DNA structure now, so let's make it to be a deoxyribose sugar. So this is a deoxyribose sugar. So if you wish to have phosphate, the 5' position is going to have phosphate. How the attachment is going on? Let's see quickly. So CH2, this will be having an O group, the OH group obviously H. Then this is having P, O minus OH. The typical structure of a nucleotide. So this happens to be a PO4 3 minus. The phosphate group. And this happens to be the nitrogenous base. Clear? So what was the question? I was asking you a question, why we are putting prime over here? The reason of putting prime. Why? Why are we putting prime over here? You know, isn't it? That DNA always replicates or DNA always extends in 5' to 3' position. PCR, obviously. Normal body DNA also. The particular action by polymerase. What is prime? See, you are having carbon over here also in the bases. And you are having carbon in the sugar also. So how to distinguish between the carbon of sugar and base? So to distinguish between carbon of sugar and bases, what we do is that we put a prime. So if you're talking about 1', 2', 3', so on, n', that means we are talking about carbon from the sugar group. And if you are only talking about 1, 2, 3, obviously n, we are talking about the carbon from the base, nitrogenous base. This is the difference we have in this particular structure. So if you are putting a prime over here, that shows that that carbon belongs to whom? That carbon belongs to the sugar. So sugar is carrying the carbon, so we are putting a prime over it. If it is present for the nitrogenous base, we will not be putting a prime over it. Clear? Just removing this out. This was just concept. I need to draw a structure over here. Okay. So if you have to attack, if you have to make the polynucleotide, what are you going to add? What are you going to add? Tell me. You will be adding one more. Let's see. This is a OH. How the bond formation takes place. We'll have a look at it. This is the OH part. So don't forget to put a OH. Put an OH on the 3' position. And here also. This makes it complete nucleotide. Deoxynucleotide. So now what happens is there is a condensation reaction going on here. What kind of condensation reaction? We are going to lose a water molecule like all the previous reactions. We will be removing this water molecule, and obviously after removal of this water molecule, what we know is we will be having a bond. So this water molecule is removed, and what we have is, as a resultant, this is the same thing we have, that is the, just moving out extra things, CH2O This 3' position will be having the O, since we are left with only a single O over there. So directly this O is going to form a bond with, see, this thing is getting removed from here. This whole thing will be removed. This will be removed. So you are going to have that O group is removed, and you have P, O minus, O, yes, CH2, O, and obviously the OH. I asked you not to miss this H. Yes, you need to add this H also. Just to add this H on the 2' position. That will show that it is a deoxyribose. Okay? So this is the structure you are getting after the whole additions and everything, the condensation and everything. So you have what? You have two kinds of bonds over here. This is one bond, is called as an ester bond, and again you are having this particular bond as an ester bond. Some of the books this is shown to be straight. I just made it a twist, but it can be straight also. So this O can be just straight as per your NCERT, don't get confused. So this CO bond and this CO bond, this is CO bond, CO bond. So what is CO bond basically called? This CO bond is basically called as an ester bond. So you have one ester bond here, and you have one ester bond here. And what you have over here, you have a phosphate group or phospho group. So these three together, when you add them all together, this ester bond, phospho and ester bond, what you get in sum total? You get a bond which is called as what? Which is called as phosphodiester bond. Isn't it? So you have a phosphodiester bond in between two nucleotides. So if you talk about two nucleotides, you have one phosphodiester bond in between them. Clear? Are we clear, everybody? Yes, children? Did you copy? All of you, did you copy this? This is phosphodiester bond present in nucleic acid. So this is what I have drawn is DNA. You can similarly draw for RNA also. What is not clear? Tell me. Yes, Shillong, what is not clear? This is condensation reaction going on. Simple. See, this is the whole molecule. This particular part is called as this PO4 3 minus is called as phosphate. Bond breaking. Bond is not breaking, bond is forming. See, I'm going to lose this whole water molecule. This water molecule will be removed. So when you remove this water molecule, what is going to happen? This HO is totally absent. Now what you have is, you have this, I'll just mark it in yellow, the things you are left with. We are left with what? We are left with this O. We are left with this P. Just look over here. Left with this O, and this thing. So what happens is, this O is present here. This is this O. Just removing this green section for a while. What What is happening seriously? See, this is the O part that you have marked in that initial substrate part, and this is the P, this is O, and remove this O. Got it? Now it must be clear. So you are removing one water molecule, as now you have been doing all the while. In lipids also we did the same thing for making an ester bond. In peptide also we did the same thing for making a peptide bond. Okay, it must be clear. It's easy. Okay, don't take it very, very seriously. It's very easy. Now you have to just do condensation, or you have to just remove a water molecule, dehydration. Dehydration or condensation reaction is going on over here to make a phosphodiester bond. Isn't it? So this is what is a particular position of DNA. I would be calling to be 5 prime. It's 5 prime, isn't it? This is what is 5. This was 5th carbon. And this is the position of DNA which is said to be the 3 prime. So whatever extension is going on in this particular molecule will move from 5 prime position to 3 prime position. So what is happening? Any new nucleotide is going to attach over here. Whatever new nucleotide you are going to attach, they are going to make a nucleophilic attack. So new nucleotides, what they do is that they make a nucleophilic attack, since you are what? You are having a phosphate, PO4 3-. So it is not having proper number of positive charges. So what it does is it makes a nucleophilic attack, and because of this nucleophilic attack, you are having the extension of DNA in 5 prime to 3 prime position. So new nucleotides are added in which particular part? New nucleotides are added on the 3-OH group. This you have studied in your 12th also. So the 3 prime OH group will have new nucleotides being added every time, and that would be what? That would be a nucleophilic attack. So you are going to form an ester bond every time that does so, and finally what you have is you have a phosphodiester bond between 2 nucleotides. Clear? So let's quickly see what's the particular thing going on over here. So one of these strands of DNA, now I did not tell you these things, one of the strands of DNA runs in 5 prime position and the other runs in this position, 5 prime 3 prime, isn't it? So what is DNA? DNA strands are anti-parallel in nature. DNA is anti-parallel, this you know. And what is the base pairing rule? A always base pairs with T and G always base pairs with C. This 3 and 2 is what? They have 2 hydrogen bonds in between, and they have 3 hydrogen bonds in between. So number of hydrogen bonds also varies. There is 2 in this structure. There are 2 in this structure and 3 in this. Isn't it? So what we have is 2 hydrogen bonds and 3 hydrogen bonds. Clear? What is this rule called? What is this rule called? Chargaff's rule. So in place of T, there would be uracil in RNA. But base pairing will not act. So we'll just can write in this way. This part will be taken by Vineet sir, how all these things are done in the molecular basis of inheritance part. Genetics part will be by Vineet sir, and he will be telling you how these particular things are actually replicating, transcripting, translating, all those things. He'll be telling you about. Okay? Clear? Tell me, children. Clear? Nucleic acid is clear? So now can you form DNA and RNA clearly? You can, isn't it? Okay, fine. So we have one more thing. We have something about the pitch of DNA as well. So let's quickly see that. Okay. So what we have is, we have two strands of DNA, one running in 5' to 3' position and the other running to 5' from 3'. Now what will happen, this particular DNA will be having, I'm drawing what, there are three types of DNA, A DNA, B DNA, Z DNA. This is what is called as the B DNA. You have to study this DNA. This is B DNA. So what B DNA has, they have is like in one single turn. This is the one single turn. Since it starts to move towards its left. From its left to another left. This whole thing from this part, you can take from right to right as well, the same thing. So this whole thing from one section, so it starts to move towards left. And after that, when it starts to move towards left another, this is said to be a single turn of DNA. So one turn of DNA has what? It has a major groove and a minor groove. Okay. And this one turn of DNA is basically given a very good name. It's called as pitch of DNA. This pitch of DNA is how much? This is 34 angstrom. How much is it? It is 34 angstrom in its total length, the pitch of DNA. This whole section from one left to another left or one right to another right has a length of 34 angstrom. Now in between them we have different bases, 10 base pairs exactly, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 10 base pairs like AT, GC, AT. You can have it anyways. This is like what I wish I'm writing. You can also do that. C, G, T, A. So what you have is 10 base pairs. So one pitch of DNA has how many base pairs? It has 10 base pairs. Mark it. It is base pairs, BP. Not nucleotides, but it is base pairs since it is double-stranded DNA. So 10 base pairs are present in one pitch of DNA. This is what is a characteristic feature of a B DNA. This is B is for biological. 34 angstrom, that means in between two bases, how many? In between two bases, how many space is there? 3.4 angstrom. In between two bases, how many space is there? Divided by 10, obviously, that would be giving you 3.4 angstrom. Done with the structure of DNA as well. Clear? All the centers, Shillong, Senapati, Tamenlong. Yes, Itanagar. Are we clear? Tell me, children. Still confusion? Done with nucleic acids? Yes. Shillong, give me a thumbs up if it's clear. Okay, everybody. Very good. Nice, very nice. Double thumbs up I'm getting. Very good. Okay, Itanagar, Tamilong. Very good, Senapati. Itanagar, just let me know, Tamilong, Itanagar, if you have any doubts. A single person doubt, 0.1% doubt also is, you have to ask. Don't carry doubts from here, because when you go back home and read, you will have doubts. Okay, nice. Very good. Yes, Itanagar child? Okay. Yes, let me know, Itanagar, if you have any doubts, or we will have a small break. No? No doubts? Okay, just let me know in after like 5 minutes if you have any doubts. We'll have a 5 minutes break. Resume after 5 minutes. Yes, children, can we resume? Okay, I was giving you 2-3 questions and then we proceed with the next topic. Clear? Any doubts with this topic? Nucleic acid, nucleotides, any doubts? Okay, this is like someone else is going on the on the computer. Good. Oh my God, not on the computer but something else is going on. Yes, any doubts? Any doubts? No doubt? Okay, very nice. So we want to give you questions. Don't tell me this was a doubt I was going to ask you. This we used to do when we were children. This was the thing we were supposedly going to ask you, ma'am, just now. Okay, fine. So, let's have 2 questions just. Nucleotide bases. Sorry. Yes. Tell me. Tell me the answer. Yes. In the meantime, I am having next question. Yes. No answers till now. Only Senapati people have answered. Yes, remaining. Shillong. What is taking so long? Quickly answer me the question. Yes, very good, Tamilong. Shillong yet to answer. All centers have answered minus Shillong. Answer this question now, everybody. Okay, done. Let's see first question, first and foremost. So nucleotide bases pair in DNA by, yes, they do pair in DNA by what? See, everybody, the pairing between nucleotide bases. These are the nucleotide bases, A, T, G, C, isn't it? And they are pairing by what? They are pairing by hydrogen bonds. Yes, Tamil clear? Am I clear? The nitrogenous bases pair by what? They pair by, always, they pair by hydrogen bonds. Okay? There is no sugar bond, there is no polar covalent bond. Nothing like that. There is only between two bases what? The hydrogen bond. And there is a particular bond which provides the stability to DNA. The stable structure of DNA is basically because of the hydrogen bond. When Vineet sir will be teaching you, he will be telling you these things also. So hydrogen bond is a basic structure that stabilizes this helix. So this double helical structure of DNA is stabilized by hydrogen bonds. Okay? So suppose your homework would be, you have to answer me 2 questions. Clear? This is clear? Can I move ahead? Everybody. Shillong, Tamilong, Senapati, Itanagar, everybody. Second question. It is asking what is the unique feature does DNA have because of its double-stranded structure, obviously. A very similar kind of questions you have in molecular basis of inheritance also. You have done your class 12th. So it must be easy for you. So it is not in all viruses. Yes, in some viruses DNA do serves as the genetic material, but it's not all viruses. And there is a variation in DNA also. When you talk about viruses, I must have taught you the biological classification that DNA, the viruses, they have 2 types of DNA, both single-stranded as well as double-stranded. So we cannot say that, yes, that always double-stranded DNA is present in all viruses. This is wrong. It is not true. It can move around the cells. No, they don't move around the cells. Never. It can act as a template. Yes, this seems to be correct. Very good. Shillong, Itanagar, nice. So this seems to be correct that yes, it serves as a template for its own copying. So when it needs to copy itself, so suppose I have a template A, T, G, C, C, G, A, A. So you would be like, if I'm talking about 5' to 3' position of this, so you can easily make the next DNA template. Why? You can just base pair it. A base pairs with T, T with A, C, G, G. Then we have is C, T, T. Obviously this will be 3' to 5' position. So this is very easy. So this is the correct answer, that is it can serve as a template for its own copying. This is what is called as DNA replication. So you have to answer a few questions. The first thing is, who proposed? Although these questions are from inheritance, molecular basis of inheritance, but yes, they're important. Who proposed double-stranded DNA structure? First question. Next, who proved? So there are 2 things, proposed and proved. Double-stranded DNA structure. I'll be getting through these questions. Who proved? So tomorrow I will be asking these things. Who proved semi-conservative nature of DNA replication? 3 questions. One more. One more? Shall I? 5 questions for tomorrow. Copied? Can I move ahead? Okay. So we classify carbohydrates under 4 categories: monosaccharides, disaccharides, oligosaccharides and polysaccharides. Mono means it is not the simplest form, not able to get hydrolyzed into further groups. So this has single unit. Di obviously means double, and oligo means somewhat, obviously from 3 to 10, few. Oligo means few. And this is many. The term monosaccharide is obviously replaceable by sugar. So we have certain differences between sugar when you talk about sugar and when you talk about polysaccharide. Certain differences you have. What differences you have? First and foremost, sugar is a small molecule, whereas polysaccharide is a macromolecule. Then this is sweet, not sweet. Next, what we have is soluble in water. This is insoluble in water. Then what we have is, this is crystalline. And this happens to be what? This happens to be, obviously, the difference, non-crystalline. Okay, this can be insoluble or this can be slightly soluble too. This can be slightly soluble also. Okay? So, this name ends with O's. Like you know some of them. Glucose, sucrose. This is basic difference between, these are basic differences between sugar and polysaccharides. Copy them and let me know if you have any doubts. Yes. Shillong, Itanagar, Senapati, Tamenlong. Doubt any? Any problem? So what is sweetest of all sugar? Sweetest of all sugar. Yes. Copied everybody? Can I move ahead? Everybody? Shillong, Itanagar, Tamenlong, Senapati are moving. So what you have is, you have certain number of micromolecules. Isn't it? And these micromolecules are what? They are called as carbohydrates. Let's go a little bit into chemistry section. So what are they? They are, carbohydrates are polyhydroxyaldoses or ketoses and their condensation products. Okay? So if it is aldoses, obviously they have a CHO group. Examples, most important of all, glucose. And if they have a ketose group, most important example, CO, that is fructose. The general formula of carbohydrate, CnH2ON. General formula where you have C, H and O in the ratio of 1:2:1. Similar to the thing you have in water also. Obviously, I'm talking about hydrogen and oxygen only. You don't have carbon in water. So, where are carbohydrates obtained from? Carbohydrates are produced during a single process of photosynthesis. So, you have photosynthesis to produce carbohydrates. Next thing we have is monosaccharides. So, they have the N part, what we mentioned in the general formula, the N part, this N part is variable from 3 to 9. So, if there is 3, that N equals to 3, they are called to be trioses. The simplest of all, the glyceraldehyde, which is simplest aldose, and we have is this happens to be simplest ketose, dioxyacetone. N equals to 4, you have is erythrose and threose. N equals to 5, that is pentose, obviously, you have ribose, you have deoxyribose, yeah, but deoxyribose is an exception to the general formula of CnH2nOn. Why? Because this has one oxygen less. Remember, we removed an oxygen molecule to make it that way. So, this is CnH2nOn minus 1. Remember this thing. This is an exception to the whole general formula. So, if we talk about general formula, this will be an exception that deoxyribose has what? It has CnH2nOn minus 1. Okay? Next thing, what we have is after that, we talk about arabinose. We have ribulose. These are all having carbon 5 in number. Okay. We can mention xylose also in this. Ribose, xylose, done. Okay. Next proceed to N equals to 6. That is basically hexoses. So the most common hexose, the most talked about, glucose. We have fructose. We have galactose. And we have is mannose. This is double N. Just rewriting this. Okay. N equals to 7. Let it be, this is not that common. Okay, it's more than enough. So you have these particular common types of carbohydrates or monosaccharides present in nature. Okay. Clear up to this, everybody? Done? Sorted? Tamilong, Senapati, Itanagar, Shillong. Are we clear up to this, everybody? Okay. So I asked you a question, which is sweetest of all sugar? Yes. What is it? Sweetest of all sugar. Fructose happens to be the sweetest of all sugar. So you take into account all sugars, you'll find. Let's have some notes. Sugar acid is ascorbic acid. It is present in vitamin C. You have one molecule called as raffinose. So raffinose is a trisaccharide. Not a di, not an oligo, but it is a trisaccharide. How is it important? Of what things? It's a trisaccharide of glucose, fructose, plus galactose. This is GALA, galactose. Okay? Then you have a molecule called as inulin. This is important. This has been given in your NCERT as well. Inulin. This is important. This is a polymer of, not insulin. There's no spelling mistake. This is inulin. INULIN. This is polymer of fructose. So fructose polymer is called as inulin. Okay. So let's now talk about the disaccharide formation. Enough of monosaccharides. Clear with everybody? Shillong, Tamilong. Are we clear up to this? Everybody? Yes. Tell me. Nice. So let's see disaccharide. Just drawing a simple structure of glucose. So you want to tell me what is this? Alpha or beta? What is this? Alpha or beta glucose? What is this? Alpha form or beta form? Tell me. Polymer of fructose, obviously in fruits. Yes. Tell me this first. Alpha or beta? If this OH group is here, this would be in alpha form. If this OH group, like suppose I'm just taking into account this thing. If this OH is over here, this is called as beta form. So this is alpha form of glucose. And this is beta form. Okay? Clear? Clear with this? Everybody? This is ring form of glucose, obviously. Inulin. This is inulin, child. INULIN. I made it clear. Insulin is a polypeptide. Why would it come over here? Okay. Insulin is a polypeptide that we have discussed as the quaternary structure of protein. That is done. Topic is done. This is inulin. INULIN. Okay? Okay. So this is alpha form, this is beta form. Now I'm going to do my equation. I'm not going to draw this thing. Did you draw that? Alpha and beta, everybody? I'll be reusing this skeleton, obviously. This is again alpha I'm having. Okay. So what we are going to have, we are going to again have a condensation reaction over here. See, lots of condensation reactions. So what is going to happen? This is like, obviously it's condensation, so removal of water molecule will take place. The similar kind of thing we are doing till now. We're going to remove this. Structure drawn, everybody? Quickly draw this structure, please. We're going to remove this water molecule. Done. Structure. Shillong, Tamilnong, Senapati, Itanagar. Clear? Move ahead. Okay, moving ahead. So what we have is, just look at this structure. You are left with what? Left with O over here. Just O is left. The red O that I marked. So this bond is going to form in this way. First carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. First, second, third, fourth, fifth, sixth. 6 carbons. This is fourth, this is first. So what we call this particular bond, this whole structure. This is what? What is this? This is C1O C4. Or we can say what? We can say COC bond. So this COC bond is what? This COC bond is glycosidic bond. So in between the disaccharides, what you have is glycosidic bond. So you are done with 4 types of bonds till now. We are done with peptide bond in between amino acids. We're done with ester bond in the triglycerides or lipids. Then we're done with phosphodiester bond in the nucleic acids. And this is the 4th type of bond called as glycosidic bond, where in the disaccharides or polysaccharides, in the carbohydrates, better we say. In between monosaccharides, either way you say. Okay? So this was what? This was the pyranose ring. This is what is called as, this structure is pyranose. The ribose thing what we drew there, over there, remember? This thing. The ribose structure. This particular thing, this was the ribose, remember? This is called as the furanose ring. This is chemistry actually. This is furanose. So if it is 5-membered ring, it is furanose. If it is 6-membered, it is called as pyranose. This is furanose. Ribose and deoxyribose, either of them. Clear? Clear, children? This is not this part, not a part of this particular section. We just made it over here just to make you understand what is furanose. Copied? Clear? Can I move ahead to disaccharides? Done this section in digestion also. Obviously 2 units are together. So first is maltose. This is maltose actually. This structure is basically what? This structure is maltose that you have drawn over here. This is maltose. This has alpha 1,4 linkage. Because the sugar is alpha, alpha 1,4 linkage. Okay, 1 to 4 like this. This is maltose. So what is maltose having? Maltose is having glucose plus glucose. Simple. We've done all these things in digestion as well. Sucrose has glucose plus fructose. Lactose has glucose plus galactose. And that's all we have. Isomaltose also used to be there in digestion. You know, isomaltose also has glucose plus glucose units. So basically they are all structures which are having similar kinds of forms. Okay? Am I clear up to this? Everybody? Shillong? Tamenglong? Okay, now you have one more thing. You have a particular term called as reducing sugar. What is reducing sugar? Now, if the Cu2 plus is reduced to Cu plus, where? In Felling's solution or in Benedict's test. So what happens is, so these particular reduction is basically having only and only, they are done by what type of sugars? They are done by reducing sugars. So copper ion Cu2 plus is reduced to Cu2 plus where? In the Fehling solution or in the Benedict test. So this particular solution is going to give you, if the color changes, okay, if the color changes, we call it to be a reducing sugar, and if not, they are said to be a non-reducing. Now you have to, all those sugars, so all those sugars having free aldose or ketose come into this category, that is reducing sugar. Okay? So if you take into account a non-reducing sugar, we have a very good example of sucrose. Why? Because sucrose has neither of these groups free. Okay, so glucose plus fructose forms the sucrose subunit. So they don't have any of the aldehyde or ketone group free for reducing the Cu2 plus to Cu plus, and thus they are not giving the particular test of reducing sugars. So they are said to be what? They are said to be the non-reducing sugars. Apart from that, lactose, maltose, all of them are reducing sugars. So this is basically, initially what we have is, if you just go into the Benedict solution or this reducing sugar concept, what does reducing sugar does is, changes the blue color alkaline solution of copper or cupric sulfate into reddish cuprous oxide. So if you are having this particular change of state, we call that molecule or that sugar to be a reducing sugar. So what happens is the blue color copper alkaline solution of the copper sulfate changes into cuprous oxide, that is obviously the cuprous oxide part, this cuprous oxide, this forms a precipitate. Okay, this pure chemistry. Okay. We're left with only polysaccharides. I guess we are running out of time today. We'll continue with polysaccharides in the next class. Along with one more topic we are left with, enzymes. We'll complete polysaccharides and enzymes in the first 1 hour. And after that I will start, let me see, it's either cell division, is it taken by Vineet sir? Has Vineet sir started with cell division? Yes, Shillong, can you let me know please? Anybody, obviously, Inchanagar, Senapati, Shillong, Tamilong, just let me know what is Vineet sir taking. Is he taking cell division? Is he taking that chapter? Yes. Anatomy? Okay, fine. No issues. Let me see. Let me talk to him once. If let's see if he is willing to take. He will take him because that comes with cell. Cell and cell division comes in a package. So let me zoology results put in the group. Put that in the group. That is the most easiest way. 5 weeks? Oh my God. That is too late. Okay, fine. Put that in group 1 style. Let's see what they tell. I will be also telling them today. You people were asking about results. Okay. I will just poke them once. No issues. Okay, so is the class clear? We are left with polysaccharides and we are left with enzymes. Okay, I'll just let them know that you people are asking about it. Don't worry, children. I will help you out. I will ask them today. Okay, yes. Tell me. Any doubts from today's class? Too much of biochemistry or biomolecules in one day, I know. No doubts? Shillong, Tamilong, Itanagar, Senapati. Do you have any doubts? Eager to have lunch? Me too. But let me know if we have any doubts or not. No doubts? Okay, great. Very nice. So just go back home and go through all these biomolecules part once, because this is a bit of different from typical biology what we are studying since like starting of the days. So biomolecules is easy. Don't panic with this. Just go with the things what I am giving in the notes form and plus NCERT. Keep your NCERT as base and you can just go ahead with these topics very smoothly. Okay, half of the people, they have opened their lunch boxes. I also need to have my lunch. Okay, thank you so much. Okay, take care. Bye-bye.